This lecture is about neuron biophysics. This is technically the last series of lectures based on the core content of the course that has to do with different topics and subfields of biophysics. Starting in the next lecture, we'll be talking about an introduction to bio-nanotechnology and a bunch of different methods that can be used as biophysical tools in different experiments and fields of biophysics. Let's start with a recap of our previous lecture. It had to do with photosynthesis and cellular respiration, as well as muscle bioenergetics. So I'm just going to go through what's the most important part for each of these different processes. Next, we're just going to jump into the nervous system and some basic anatomy of neurons. Once we understand what the neurons look like, what different types of cells there are, we're then going to take a look at neuron action potentials. Basically go through the mechanism of an action potential and signal propagation in the neural network. Afterwards, we'll take a look at some experimental neurobiophysics, which has to do with how we as experimental biophysicists can apply different biophysical tools to analyze action potentials as well as different signals from the brain. So now let's start our recap. We're first going to go through some redox reactions, basically go through what they are, then go into photosynthesis, the main steps involved, as well as cellular respiration. First of all, we know that ATP from the previous lecture is a high energy molecule that stores energy for most biological processes. Cells will actually conserve energy in the form of ATP by coupling its synthesis to the release of energy through redox reactions or oxidation reduction reactions. So in a redox reaction, what happens is an electron is passed from a donor to an acceptor. The oxidation of a molecule refers to the loss of electrons, whereas the reduction of a molecule refers to the gain of electrons. If you've taken some chemistry courses, you'll know that the very common way to remember this is oil rig, where oxidation is loss and reduction is gain. A molecule being oxidized acts as the electron donor, while a molecule being reduced is going to act as an electron acceptor. Since the electrons represent energy over here, a substance that has many electrons that can be donated, such as compound A, is thought to be energy rich, which is where ATP comes in. Now let's look at a grand overview of photosynthesis. You have the light reactions and you have the light independent reactions. So what's important here is that this is a chloroplast cell in plants. The light dependent reactions are going to absorb light energy in the thylakoid membranes and then take in water and then expel oxygen at the very end. Now let's take a look at the specific details of the light-dependent reactions in the next slide because there's too many to cover just in this one. But then you also have the Calvin cycle taking place after you get ATP and NADPH from your light-dependent reactions. Through the Calvin cycle, you'll then be able to take in carbon dioxide and then make sugar after converting ATP and NADPH back to ADP with a phosphate group and NADP plus. So it's a constant cycle of photosynthesis. Now let's take a look at the light-dependent reactions within photosynthesis. So in step 1, you have light absorption in photosystem 2. Light is absorbed and passed inward from coherence from pigment to pigment until it reaches the reaction center. The reaction center is labeled P680 because it absorbs that wavelength, exciting an electron. So that electron gets excited. It's then passed to an acceptor molecule and replaced, and that's replaced with an electron from water, and that releases oxygen that we breathe out. Next, you have ATP synthesis happening over here. So electrons from step 1 are going to travel down the electron transport chain, where the released energy drives a proton gradient. Basically, you have H plus from the stroma to the thylakoid interior. Your protons are going to flow down the proton gradient back to the stroma and pass on and pass through ATP synthase to spur ATP production. And this specific process, which is important to know, is called chemiosmosis. So all of that produces ATP through ATP synthase. So step 1 and step two allow for light absorption and the creation of ATP. Now, there's another light absorption event that takes place, and that's in photosystem 1. So what's going to happen is there's still an electron that's going down this electron transport chain. It reaches photosystem 1 and joins with photo reaction center P700 with a chlorophyll A. Light is absorbed over here, and then another electron is available to go through another electron transport chain. That's step number 4, where this electron, instead of creating ATP, creates NADPH. So that covers the light-dependent reactions. Next, you have the Calvin cycle. So once you have that ATP and the NADPH from the light-dependent reactions, it moves on to the Calvin cycle. And the first step in the Calvin cycle, stage 1 over here, is called carbon fixation. In the presence of carbon dioxide and rubisco, what happens is carbon is fixed from an inorganic form to organic molecules as 3-PGA. There's actually 6 in total of 3-PGA that are created. And then this is going to, in step 2, be reduced. And ATP and NADPH are going to be used to convert 6 3-PGA into 6 molecules of G3P. So we're up to here now. Let's see what else we have. One thing is, after the ATP and the NADPH are used to create the G3P, the ADP and the NADP plus are going to be reused and re-energized for the Calvin cycle. The last step, once you get over here, you'll end up with the regeneration of RuBP. Some G3P molecules are going to go make glucose over here, and then some are going to go back to be regenerated to restart the Calvin cycle. So you have some that go for glucose and some that go for regeneration. And that's probably the most important part of step 3, the fact that there is a choice that G3P has to make, either go back into the Calvin cycle or be used for the creation of sugars. Okay, so now that we have gone through a really brief overview of photosynthesis, we do need to go through another overview of cellular respiration, pretty much the polar opposite of photosynthesis. So in cellular respiration, there are 3 main steps. In our original lectures, we talked about 4 main steps, but we also mentioned that step 2 that we had, pyruvate oxidation, could be considered a transitional stage. So we're now going to combine it so that we have a shorter overview. So step 2 and 3 in our previous lectures is now step 2. So in step 1, glycolysis occurs. It occurs in the cytoplasm. So right here, there's glycolysis. It occurs in the cytoplasm where a 6-carbon glucose molecule, which is basically the broken down food that we've eaten earlier, is further broken down by enzymes into pyruvic acid or pyruvate. The execution of this process usually takes 2 ATP and produces a net gain of 2 ATP. So step 2 is called, where am I? Step 2 over here, the little blue arrow, step 2. We combine pyruvate oxidation and the Krebs cycle together. Pyruvate is converted into a 2-carbon molecule that's bound to coenzyme A. That creates acetyl-CoA. And then the Krebs cycle itself actually begins when that acetyl-CoA combines with a 4-carbon molecule called OAA. This produces citric acid, which has 6 carbon atoms. This is why the Krebs cycle is also called the citric acid cycle, since citric acid is created. After that, it goes through a series of reactions that release energy, of course, ATP, as well as NADH. And finally, there's also a third one, FADH2, that's an energy carrying compound that's created. So it's very important to know that in cellular respiration, after the Krebs cycle, you end up with not just ATP and NADH, you also end up with FADH2. So there's 3 different types of energy carrying compounds that are created from the Krebs cycle. Of course, because of this cycle and the creation of energy-rich molecules, there has to be some sort of product that's released, and that's going to be carbon dioxide, which is the waste product of these reactions. Okay, so step 3, which is commonly known as the electron transport stage, is another term for oxidative phosphorylation. High energy electrons are released from NADH and FADH2, and they're going to move along an electron transport chain, just like those used in photosynthesis that we talked about before. As those high energy electrons are transported along the chains, some of their energy is captured and it's used to pump hydrogen ions or protons across the inner membrane from the matrix into the intermembrane space. What happens is the pumping of the protons in the intermembrane space are going to create a chemiosmotic gradient and cause the ions to flow back into the membrane, and that is going to allow for ATP synthase to act as a channel protein to form ATP from ADP. And this is where you end up with oxygen being created as well as water. Sorry, just to confirm, you end up with a carbon dioxide created from the first stage, and then in this stage you create water because oxygen is required to form that water. Okay, so the last part that we talked about was ATP and muscle bioenergetics. I have a new picture over here taken from here. And I think it really gives a good overview of the different steps that we have to know about in terms of muscle bioenergetics. So just remember, you do have to know about how ATP plays a role in the contraction of a muscle. We did take a quick look at it when we talked about muscular biophysics at the beginning of the term. Now it's really important that we understand how that ATP plays a role and where that energy comes from. So muscles are going to contract in a repeated pattern of binding and releasing between 2 thick and thin strands of the sarcomere. ATP is absolutely critical to prepare myosin for binding and to recharge the myosin. So I have myosin over here. ATP is first going to bind to myosin, not from step 1, but in step 4. I want to start here because it's a little bit easier to explain when ATP first attaches. So if you're asked a question about how does ATP play a role in muscle bioenergetics, then this is probably where you would start. ATP first binds to myosin, moving it to a high energy state. The ATP is then hydrolyzed in step 5 into ADP right here and an inorganic phosphate by the enzyme ATPase. The energy released during ATP hydrolysis changes the angle of the myosin head. So take a look over here. The angle of the myosin head is kind of backwards, but after it's hydrolyzed, the myosin head is kind of in the forward direction. So this is called the cocked position for a myosin head. It's now ready to bind to actin if the sites are available. ADP and your phosphate remain attached until later on. And now this myosin is considered in a high energy state. So now we have 5, we go back to step number 1. The muscle contraction cycle that we talked about previously at the beginning of the term starts at stage 1, but that's not when ATP starts binding. So the contraction starts at stage 1, but the ATP starts at stage 4. So in step 1, we have the beginning of the muscle contraction cycle. It's triggered when calcium ions over here bind to the protein complex troponin. so right here, it exposes the active binding sites on the actin filament. Now, as soon as the actin binding sites are uncovered, right here, once that's uncovered, we move on to step 2. The high energy myosin head is then going to attach to and create a cross bridge with that actin filament with its exposed binding site. Once the myosin binds to the actin, the phosphate is then going to be released, as well as the ADP. And because those are released, it's then going to go back to its original position over here and go backwards. So once the myosin binds to the actin, the ADP and the phosphate are released, and the myosin is going to undergo a conformational change so that it goes back to its original energy state. As myosin expands the energy, it moves through what's called a power stroke, which pulls the filament towards the M line. And when the actin is pulled approximately 10 nanometers towards the M line, the sarcomere is going to shorten and your muscle is going to contract. And at the end of one power stroke, the myosin is now in a low energy position. You then have ATP coming in to attach to the myosin again, which allows the whole muscular contraction cycle to restart and continuously contract your muscle until it stops. So hopefully that gives you a good overview of, or a good summary of what we talked about in the previous lecture. Now let's talk about the nervous system and neuron anatomy. This is one of the most unique chapters of the course. I've done research in it, and as a past PhD student and postdoc of Professor Zoylianenko, we do focus a lot on Alzheimer's disease, as well as using biophysical tools to take measurements of different proteins and ions that will affect Alzheimer's disease. So the nervous system is truly complex because it helps coordinate your body's actions and sensory information by transmitting signals to and from different parts of your body. And in fact, the nervous system usually detects changes and then coordinates with your endocrine system to respond to those changes. To start understanding the nervous system, we do have to break down this intricate network of communication into 2 sections, and that's the central nervous system and the peripheral nervous system. The central nervous system consists of your brain and your spinal cord, and it acts as the control center of the entire system. It helps coordinate all your reactions and influences the activity of all parts of your body. Meanwhile, you have the peripheral nervous system. It consists mainly of nerves, and these nerves are basically bundles of long fibers or axons that connect the central nervous system to every part of the body. So you have the central nervous system being the brain and the spinal cord, and then you have the peripheral nervous system being the messengers that carry those signals across to the rest of your body. And because of that, the peripheral nervous system is just so much more complicated than the CNS. So we have to divide the peripheral nervous system into 2 more parts. What goes to the CNS and what comes from the CNS. So you have the sensory or afferent version of the PNS that contains nerves that transmit information from the body to your central nervous system. And you have the motor or efferent division of the PNS, which contains nerves that transmit information from the body from the brain to the rest of the body. So very important to know the differences between the 2 of these. Now, for the purposes of this lecture, we're just going to focus on the motor or efferent division of the PNS because it has to do with what we can control and what we can't control. So let's now focus on the motor division, and we can further divide that into 2 parts, the autonomic and somatic nervous systems. So usually when in most biology classes or human anatomy classes, when you talk about the peripheral nervous system, it's usually just broken down into the autonomic or somatic nervous systems, because even though it can be broken down into two different divisions, it's commonly known as two separate systems. So the autonomic nervous system is what I'll start with first, because we want to focus on the somatic nervous system for the rest of the lecture. The autonomic nervous system controls our involuntary motion, means we have no control over it. The CNS sends impulses to our heart muscles or cardiac muscles, our smooth muscles, and our glands in our bodies, just to help us regulate our entire body without our knowledge. And there are two types. You can have the sympathetic division or the parasympathetic division. Very important to know about these two. The sympathetic division activates in cases of emergency. So this would be our fight or flight syndrome. Basically, if you have an emergency, the sympathetic division is going to kick in to give us energy to react to that emergency. You then have the parasympathetic division. It helps us conserve energy, so the opposite. Basically, when we're in a resting state or when we're sleeping, this is the division that will help us conserve energy and promote the housekeeping of our bodies, just so that we can save energy for another time. So that covers the autonomic nervous system. We now go back to the somatic nervous system. And the somatic nervous system governs voluntary motion through skeletal muscles. And this is what all of our background and all of the lectures lead us to, because the somatic nervous system consists of sensory nerves and motor nerves that are going to promote muscular and help us control muscular contraction. Let's go to the next slide. So in the somatic nervous system, I do want to explain something called the reflex arc, which comes up quite often in human anatomy and in medical sciences. But first, a quick recap of the somatic nervous system. It's the part of the peripheral nervous system associated with the voluntary control of bodily movements through skeletal muscles. There are afferent and sensory nerves that relay the sensation from the body to the CNS. And there's afferent motor nerves that send out not comments, but commands from the CNS to the body and stimulate muscle contraction. Now, there's something called voluntary reflex arcs. So when a stimulus A is encountered, you then have a signal being sent from that stimulus all the way up to C, the spinal column. So B represents that stimulus signal. D is here in purple to represent an interneuron. Basically, instead of going up to the CNS, sometimes you have a signal that's passed through an interneuron that connects to E, which is a motor neuron. And then that motor neuron is going to fire a signal off to the muscles, which then contract in red through F. And then G is just the label for the bone. Basically, the muscle contracts towards the bone. So this whole thing from the stimulus to the contraction is called a reflex arc. And it's very important to know about a reflex arc because not every action or not every voluntary motion we have is going to go directly to our brains and then back. Since the action potential doesn't travel to the brain in some cases, it allows us to have faster reflexes to certain types of stimuli. So a good way to express this is that the purpose of the reflex arc is so that we can bypass a signal traveling to the CNS and back in order to create a reaction. Sometimes we do want to activate spinal motor neurons without the delay of routing signals through the brain. The brain is still going to receive the input, but the action can be carried out faster. Okay, so now that we know about the different types of systems within a nervous system, we want to focus on what kind of cells are in the nervous system. And essentially, there's only two types of cells. There's neurons and there's glial cells. So neurons are electrically excitable cells that communicate with other cells through synapses. They're the main component of nervous tissue and can be classified by structure or function, and we'll go through those in a bit. A neural circuit is created when a group of neurons are connected. Now you have a glial cell. These are non-neuronal cells that provide several different functions that you'll have to know about. Support and nutrition. They help maintain homeostasis. They form myelin for the myelin sheath to allow for saltatory conduction to occur, which we'll talk about later as well. And they also participate in signal transmission. So these two are kind of connected together. But the most important thing about neurons is that they pass on electrical signals along their axons. So here, a typical neuron usually consists of a cell body right here, which is usually called the soma. It's got dendrites and a single axon. This is a typical neuron. The soma is usually really compact and consists of a nucleus and some other organelles. There's a bunch of dendrites here that extend from the soma, and each dendrite might even have more dendritic branches that can travel several hundred micrometers away. You also have an axon that leaves the soma through the axon hillock, and this is a more bulgy part of the soma. And this axon can actually travel as far as 1 meter in a human body to its axon terminals or synaptic terminals, where it will leech onto another neuron through its dendrites. But this isn't what every neuron looks like. This is just a typical look at its structure. There are some things missing, which we'll discuss later, which are the myelin sheaths that cover that axon. But first, I just want to go through the anatomy. How do we categorize different neurons? We use the number of dendrites or extensions from the cell body or the soma to determine what kinds of structures we have for a neuron. We have unipolar, which means here is our cell body. You can't really see any dendrites here, and you only see the axon coming at one end, and then a bunch of axon terminals. So this would be the simplest type of neuron, unipolar. You then have bipolar, where essentially you have two extensions on opposite sides of the soma. You still have one axon, but the soma is located in the middle of that axon, and one dendrite over here potentially to receive signals. And then you have your axon terminals here. So in other words, your cell body has kind of migrated to the center of your neuron. Now you have pseudo-unipolar, which means you have your cell body which receives the signals. It goes down through the axon, but somewhere in the middle, the axon splits into two, and you end up with two sets of dendrites. You then have multipolar, where you have a cell body, and this is the one that we presented before. You have a bunch of dendrites here, which receive signals, go down an axon, and then go to the axon terminals. And these are the four most basic types of neurons by structure. You actually have many, many more. These are just typical ones. There are some very unique looking neurons throughout our body as well, and some examples are provided in this slide. So these unique neuronal types are usually specific to their location, and they come with a distinct shape. So you have dendrocytes, ovoid cells, Purkinje cells, many, many more. One example I want to focus on is the Purkinje cells. These cells are really huge and kind of fibrous looking. They're found in the cerebellum and are very large. So it's specific to the function over there because the cerebellum controls many things in your body. So this over here is specific to your cerebellum. Now, another one, maybe you have specific cells that need to travel across your brain, so they have to be longer. The one that looks the longest over here would be this one, a spindle-shaped cell. So these are very long and branched in a unipolar direction, and they usually help connect widely separated areas of the brain so that they can communicate with each other. So because you have all these different structures, this leads us to a discussion of the different functions of neurons. Because there are so many shapes, we can't guarantee that we'll always look at a shape that has a specific function. So sometimes we categorize neurons by their function instead of their structure. So what kind of functions are there? There are three main functions if we look at signal direction, but if we look at function, it can have many subsets as well. So not only can we determine the function, or determine the type of neuron by the signal direction, we can take a look at the discharge patterns of the electrical signal. We can take a look at the neurotransmitters that are released for specific neurons and just have an entire category based on, let's say, acetylcholine neurotransmitter releases, so on and so forth. And of course, you can also have neurons classified by their actions on other neurons. So that would be what kind of action that they produce after they send that signal to another neuron. For the purposes of this course, we're going to focus only on signal direction. So there's only three types that we have to care about. There's afferent or sensory neurons. That would be the neurons that convey information from your tissues back to your brain or the CNS. You have efferent or motor neurons, which transmit signals from your brain to your body. And you have interneurons, which were really important in those reflex arcs. They connect neurons within specific regions of the CNS. And basically that means within the CNS, it includes the spinal cord. That's where most of those interneurons would be. So we've covered neurons. Now we move on to glial cells. There are many types of glial cells, but they have four main functions. They can surround neurons and hold them in place. They can supply nutrients and oxygen to neurons. They can insulate neurons from one another. They can also destroy pathogens and remove dead neurons. So because of this, they're considered the glue of the nervous system, just because they provide support in so many different ways. Glial cells can be separated into macroglia or microglia. Over here, all of these are macroglia, and you have microglia as a separate subset, which we won't focus on. We will be focusing on macroglia, specifically Schwann cells. In the CNS, you have many different types: ependymal, oligodendrocytes, and astrocytes, whereas these commonly get mutated and cause brain cancer and brain tumors. But in the peripheral nervous system, there's two types: satellite cells and Schwann cells. We're going to be focusing on Schwann cells because they play a huge role in neuronal action potentials, as well as saltatory conduction. What they do is they help form a myelin sheath around the axon of motor and sensory neurons. So you can imagine this purple rod over here is an axon, and the yellowish stuff would be the myelin sheath that coats that axon, with this being the Schwann cell that creates the myelin that creates the sheath. So now that we have a structure of a neuron and the structure or the contribution of the glial cells, specifically the Schwann cells, we can have a more detailed diagram of our typical motor and sensory neuron. So a typical motor and sensory neuron would then have your Schwann cells creating a myelin sheath. There are many, many myelin sheaths along the axon, but in between each of these sheaths is a gap called a node of Ranvier. So it's important to understand what is happening in the axon in terms of the contributions from your Schwann cell. In this section, we'll be taking a look at neuron action potentials. So that's basically how neurons send signals to other neurons and create a chain of electrical signals that pass on from one neuron to another in a neural network. We're going to go through an overview of a membrane potential and why it's even there in neurons. Then we're going to take a look at how that potential is created and different ways to take a look at it. Then we're going to take a look at a detailed mechanism of how an action potential occurs through saltatory conduction. First, we take a look at how a membrane potential is created in neurons. Neurons are going to send messages electrochemically. Important ions are sodium, potassium, calcium, and chlorine, with some negatively charged protein molecules. Neurons are surrounded by a semi-permeable membrane that are going to allow some of these ions to pass, while blocking the passage of some other ions. Since the membrane, as we know from our lipid membrane chapter, are asymmetrical and semi-permeable, that already in and of itself creates a membrane potential. And it can be described by several different types of potentials that we need to know about. These three potentials will be covered in the next few slides. Very important to know what they mean and how they play a role in calculating membrane potential. The Nernst potential, which you know about already, covers the diffusion of ions because of the differences in ion concentrations from the outside and the inside of the membrane. And that comes because of membrane asymmetry. You have what's called the GHK potential, or the Goldman-Hodgkin-Katz potential, usually representing the potential calculated from the Goldman equation. That takes into consideration the different membrane permeabilities for each ion to determine total membrane resting potential. So think about the Nernst potential as just being able to calculate the diffusion and the potential, think about the Nernst potential as the potential from the diffusion of one ion, or basically contribution of one ion. If you take into the contribution of all of the ions as well as all of their permeabilities, you end up with the Goldman-Hodgkin-Katz potential. So that's the main difference between the two. One takes into account everything, one takes into account the ions one by one. You then have the Denon potential, which we talked about before, comes from the Denon equilibrium. Basically, your membrane has different permeabilities to different substances, such as large charged proteins. And those different permeabilities will also be covered in the Goldman-Hodgkin-Katz potential. So the Nernst potential develops due to the division of particular ions across the membrane. If your ionic concentrations inside and outside are different, the fusion potential develops. So this equation right here shows us that the potential across the membrane is predicted by 5 relationships, which I'll list in a bit and are important to know. First of all, in equilibrium, we know that the ion current has to be 0. So that means the concentration gradient is going to be balanced by a diffusion potential through the Nernst equation. So if your ion current is 0 and the flux is 0, it means that there has to be a potential that's maintained across the membrane, because one side is going to have a higher concentration of an ion versus the other side. So the Nernst equation is this. We've seen it before. I don't have to go through it again. But what we can do is use the Nernst equation to calculate the electric potential across the neuron's membrane. But the 5 relationships I want you to know about is what the potential is proportional to. So the potential here is related to the ideal gas constant R, because the motion of ions is involved. The potential is going to be proportional to the absolute temperature T, because ions are going to move faster at higher temperatures and slower at lower temperatures, of course. The next one I want you to know about is that the potential, because we know that ions diffuse from a region of higher concentration to a region of lower concentration along a concentration gradient, the diffusion happens along a concentration gradient here. Now we take a look at what it's proportional to again. Potential is going to be inversely proportional to the charge of the ion, called Z, because one needs fewer ions to yield the same difference in charge if each ion has a greater charge. Now, potential is also inversely related to F, Faraday's constant, which is a unit of electrical charge, which has to do with the ion's charge as well, due to the same reasons. So those are the 5 different relationships that will help us predict the potential across the membrane. Here's a practice problem that you can do to calculate the Nernst potential for several different ions given the following concentrations in millimolars per liter. The inner concentration is given and the outer concentration is given for each of these ions. Calculate the Nernst potential for each of these separately. And this would be very good practice for questions that come up. Now, moving on to the Goldman-Hodgkin-Katz potential, I present the Goldman-Hodgkin-Katz voltage equation because that's where the potential comes from. It's commonly known as the Goldman equation. It's used in membrane physiology to determine the Nernst potential across a cell's membrane. But what's unique about the Goldman equation is that it takes into account all of the ions that are permeant through that membrane as well as their permeability constant. So this is the equation if you have sodium, potassium, and chlorine. In the steady state GHK condition, you do have an overall net flux of 0. So the individual ionic currents exist independently, but the total current is 0, so your net positive flux must equal to your net negative flux. So the condition for GHK is that you have all your positive flux equaling your negative flux. So JK plus JNA is going to be equal to JCl. So using that basis and that equation, try this practice problem. Calculate the GHK potential for a neuronal membrane, assuming the values from problem 19.1 give you the inside-outside concentrations for a neuronal membrane. Some additional quantities you might want to know of. Assume the temperature is 20 degrees Celsius. Assume that P represents the membrane permeability for each ion, which are given by this relationship. pK is 1, PNA is 0.05, and PCl is 0.45. R is the universal gas constant, and F is Faraday's constant. And what I'd want you to do is make sure you do a unit analysis so that you can find out what the potential, the units of potential are. So use that as practice as well. Next, we have the Denon potential. We've talked about the Denon equilibrium before. The Denon potential comes from the fact that large molecular ions that can't move across the membrane are going to be concentrated inside the cell and contribute to an electric potential across that cell membrane. And because that creates that Denon equilibrium, if that equilibrium exists, that means the membrane potential exists at which the ion movement into the cell is equal to the ion movement outside the cell. So the ion movement is equal, but the charge is not. So what does this mean? And if there is a Denon equilibrium, does it mean that there's a resting potential? No, because in the Denon equilibrium, all of the permeable ions are in equilibrium across the membrane, but still provide a membrane potential. There is thermal equilibrium, there is electrical neutrality, and there's also the conservation of ions. But that is not true for a resting potential. So very important to know what the difference is between a resting potential and the other potentials presented here. And we'll go through that once we talk about action potentials. Now, practice problem 19.3 goes through the Denon potential. You're trying to calculate the Denon potential that develops across a membrane given all of these values. So use the Nernst equation and potentially the Goldman equation. Try and figure out what kind of Denon potential difference develops across the membrane from this over here. What you'll have to do is first take a look at the conditions for the Denon potential, basically thermal equilibrium, electrical neutrality, and conservation of ions. You'll have to make some assumptions about the concentration of ions over here, and then you'll be able to plug them into the Nernst equation to calculate the Denon potential. So take a stab at that, and we will go through the solutions in one of the live lectures. Now, next we want to take a look at, or start to take a look at nerve impulse propagation. So that starts our our little exploration into how signals are passed from one neuron to another neuron, starting from the resting potential. So nerve conduction and stimulation is based on ion exchange and the potential change across the membrane. Your resting potential is known as approximately 70 millivolts. The change in the ion concentration or the electric field as a response to stimulus leads to membrane depolarization and nerve impulses. So I'll go through all of that in a lot more detail in a bit. If your membrane is depolarized, an action potential is going to take place until a hyperpolarization event results in overshooting that threshold and allowing it to slowly return to its resting potential. So here, what's happening is you have a difference in the concentration of ions from each side of the membrane. In the resting potential, there is going to be a potential of negative 70 millivolts that's maintained across the membranes, especially if you take a measurement. And you should be able to calculate that too from some of those equations that we've given. In a depolarization event, you're going to have a bunch of sodium ions flow in, and therefore it causes a depolarization of the membrane down to approximately 0 millivolts, for example. Completely depolarizes. A hyperpolarization event is when, for example, your potassium ions leak out and you end up with a bunch of potassium ions on the outside and a bunch of sodium ions on the inside. In this case, it's hyperpolarized. Now you have too many potassium ions outside, so now you have a plus 40 millivolt reading. And in that case, what will happen is it will slowly go back to resting potential. So this summarizes all of it in a graph, which is much better. First, we want to define what an action potential is. An action potential is a membrane potential of a specific cell location, and it rapidly rises and falls. As it rises and falls, a cascade of depolarization events follow that action potential, so a signal gets passed on. What's very important to know is that action potentials are all or none. That means it either happens or it doesn't. You don't get a weak signal and you don't get a strong signal. You just get one steady signal. So if it passes a threshold, there is an action potential that takes place. If it doesn't pass the threshold, it doesn't take place. In neurons, these action potentials are absolutely critical, and they play a central role in cell-to-cell communication through what's called saltatory conduction. So after we talk about an action potential, which is basically what happens within one part of the axon, we then take a look at saltatory conduction, which allows for the transfer of that action potential along the entire axon of your neuron. So taking a look at this graph, this is the approximate plot of a typical action potential, which shows its various phases from steps 1 to 5 and on repeat. So the membrane starts at minus 70 millivolts over here at a time of 0. So that's its resting potential, number 5. A stimulus is applied in stage 1, and that stimulus raises the membrane potential above negative 55 millivolts. So that's the threshold for the all or none action potential that you'll have to remember. So the stimulus is applied. If it fails, nothing happens. If it goes past, then a depolarization event occurs. This depolarization event, what's going to happen is it rises to a peak potential of plus 40 millivolts because of a huge influx of sodium ions. And this takes approximately like 1 millisecond. Just also in another millisecond, the potential then drops and overshoots to a potential of potentially negative 90 millivolts. And finally, this is a relaxation period where the hyperpolarization event dies down and the membrane potential goes back to its resting state. So all of this in action potential takes place within 4 to 5 milliseconds. Now, for the mechanism of an action potential, these are the steps that you should be familiar with. There were 5 steps that we presented before. Step 1 is a stimulus generating a potential. The stimulus is going to initiate graded potential large enough to pass the threshold and open the voltage-gated sodium channels for an influx of sodium ions. Step 2, there's a rapid depolarization after it overshoots to plus 40 millivolts. Sodium is going to rush in, cause rapid depolarization, and the inner membrane becomes more positive. That means the transmembrane potential of plus 30 millivolts closes your voltage-gated potassium channels and moves them out to shift the potential back to resting levels. There's a repolarization event. The membrane continues to move towards resting levels, and the sodium and potassium channels stay open. In the hyperpolarization stage, step 4, the potassium channels remain open, the sodium channels close, so the inner membrane now becomes more negative 90 millivolts. In step 5, you have the resting potential. The voltage-gated potassium channels are going to close, and the potential returns to normal because of the sodium-potassium pump. So that's just a very basic overview of the mechanism of an action potential. Now for the mechanism of saltatory conduction. First, we have to define what saltatory conduction is. It's the propagation of action potentials along myelinated axons from one node of Ranvier to the next. So in between each of these myelin sheaths made by a Schwann cell, you have a node of Ranvier, and what happens is when your action potential reaches here, it's going to jump and cause another depolarization event to initiate another action potential. And this gets passed on all along the axon until the entire signal reaches the axon terminal to release neurotransmitters. So myelinated axons are only going to allow action potentials to occur at unmyelinated nodes of Ranvier that occur between these myelin sheaths. This restriction is really important because it allows the action potentials to propagate at a very rapid speed. Instead of propagating at every single incident here, you can have an action potential jump from one node to another node, which speeds up the whole process of signal propagation. Taking a deeper look at what happens during an action potential and signal propagation, sodium is going to rush into the node, and it creates an electrical force that pushes on the ions already inside the axon. The rapid conduction of that electric signal reaches the next node and creates another action potential, thereby refreshing the signal. So it's basically the rapid opening of the sodium ions and the rushing in of the sodium ions that creates the electrical force that allows that signal propagation to occur, because this is the next node that is free for those sodium ions to travel to. So saltatory conduction allows electrical nerve signals to be propagated long distances at very high rates without the loss or degradation of the signal. There is no degradation of the signal because it's all or none. Either it's a depolarization event that triggers an entire action potential, or there's none at all. There are also several stages for neurotransmission. Basically, now that we've talked about an action potential taking place at one specific node of Ranvier, you then have a bunch of action potentials being passed on and on until it reaches the end of your neuron at the axon terminals. At the end of your axon terminals, you have a synapse. So at the end of the axon terminal, where it kind of touches the other dendrite or the receptor, you have your presynaptic neuron over here, the first stage that takes place is the synthesis of the neurotransmitter. This can take place either in the cell body and have it travel down to the axon terminal, or just be in the axon terminal itself. Then you have the storage of your neurotransmitter somewhere in here until it's required. It's going to be stored in storage granules or vesicles in the axon terminal. What usually happens is calcium enters the axon terminal during an action potential, and it causes the release of those neurotransmitters into the synaptic cleft right here. So that's the synaptic cleft is the little space or the gap between the axon terminal and the next neuron's dendrite. After the calcium is released, after the neurotransmitter is released, these little dots over here, the transmitter is going to bind to and activate a bunch of receptors in the postsynaptic membrane. And that's going to cause another depolarization event that passes on the signal. The neurotransmitter in the final stage is going to be deactivated. It's either destroyed by enzymes in the synaptic cleft, or it's taken back into the axon terminal from which it came, where it can be reused or degraded and removed. So the last few slides are about experimental neurobiophysics. There are so many more things that I can talk about regarding these techniques. I also have a lot of experience with Alzheimer's disease in Professor Zoilianenko's lab. So for anybody who's interested in learning more about neurodegenerative diseases, or just how we can apply different biophysical tools to the study of different neurodegenerative diseases or different systems within your brain, detecting brain signals, definitely speak to myself or the teaching team because we're all familiar with it. The two that I want to focus on for this lecture are ion current recordings through patch clamp, as well as electric analogs of neuronal membranes. Okay, so first, we want to talk about what neurobiophysics is. Neurobiophysics is the subset of biophysics that focuses on, specifically through a definition from the Biophysical Society, is the development and use of physical techniques to gain information about the nervous system on a molecular level. From Wikipedia, common description is the application of experimental biophysics to study the nervous system. So these are common explanations of what neurobiophysics is: the application of a biophysical tool to study something related to the signals within your brain and your body. So we want to focus on how we can take these measurements. If we're trying to focus on getting information about the nervous system on a molecular level, then we have to find a way to detect excitations within neurons, as well as how those signals are passed, or how information is passed from neuron to neuron. So you end up with these two focuses. So in the nervous system, we focus on electric signal propagation, and we can detect brain activity using various recording techniques. So there's two sets of experimental biophysics that can take place when we try and record signals. Number one, you can take a look at the molecular level and look at membrane potential changes through ion current recordings. That would be a in-lab technique. You can also do EEGs, which detect signals from within your brain that are covered in the methods lectures that are posted, as well as in more detail in future courses. So in terms of an ion current recording through a patch clamp, what is a patch clamp? A patch clamp is a glass pipette, which you can see over here. It has a really tiny opening, and what happens is it's used to suck up a little piece of your membrane, like this, to measure the membrane current through single ion channels. So there's clearly going to be some sort of extracellular solution over here, and there's going to be an intracellular solution over here that can be used. But this picture shows three different ways that we can take measurements. It presents 3 different types of patch clamp measurement techniques for ion channel activity. And the red dots here are just using acetylcholine as a sample neurotransmitter. In the cell-attached mode, which is A, right here, and you'll have to know these names. In the cell-attached mode, a pipette is pressed tightly against the cell membrane. You apply some suction, and a little piece of the membrane gets sucked up. And by doing that, a really tight seal is formed between the pipette and the membrane. The seal is going to ensure that the pipette is going to capture the current flowing through the channel. And in the cell-attached membrane patch over here, the intracellular contents remain undistributed. Let's see. So in this picture over here, the intracellular contents originally had the neurotransmitter, and the acetyl. Wait, let's see what happens. Right. Okay. So the acetylcholine is already in the pipette. That's the solution that's in the pipette. And then you have your neuronal membrane over here. You have these potassium channels that are sensitive to acetylcholine. So if they sense the presence of acetylcholine, they will open, and then you will have a gate that opens up, and you can detect the conductance across the membrane. So that's one example. Now next, you have the inside-out mode, panel B. So after a cell-attached mode is created, what you can do is just completely rip off the bottom of the membrane, so you have this floating piece, but the protein is still here, or the ion channel is still here for us to manipulate. So there's going to be a brief exposure to air, and it only disrupts the free hemisphere of the membrane, leaving the formerly intracellular surface exposed within the next solution that you bathe it into. Now, what happens is you have the cell membrane, but what you can do is modify the intracellular contents and the extracellular contents. So you have full control over each side of the membrane to perform specific studies on, to check out the ion current that takes place from different substances. So there's these 2 modes. Then there's called what's the whole cell mode. In the whole cell mode, you have the pipette that sticks in, and it ruptures right here. So you have a cell-attached patch, starts from here once again, and a pulse of suction from the pipette bursts the membrane at the very top, but the suction is still there. It's still attached to the whole cell. So now the entire intracellular cell is accessible to the pipette. So this means that you can create a larger barrier for interaction with your solution. So these 3 different ways, just various methods where you can manipulate the intracellular or extracellular environment, as well as manipulate the type of focus of your experiment. So how do we record voltage? There are 2 ways in which this can be done. There's the voltage clamp technique, and there's the current clamp technique. And simply put, we're either holding the voltage constant or we're holding the clamp constant, the current constant. So in the voltage clamp technique, the voltage across the membrane is controlled by us, the experimenter, and the resulting currents are recorded from the changes that take place. So there's an electronic feedback system that's in place. There's going to be a measured potential that's caused by the change, and it's always going to revert back to its original potential. So it's a measured potential that's held against the holding potential to determine the current that can create this potential. Now you have the current clamp technique. This is when you have a current passing across the membrane, and that current is controlled by us, the experimenter. and the resulting changes in voltage are recorded. So these are generally in the form of action potentials that we want to record because we've sent a current through the neuronal membrane. Now, I won't discuss too much of this, but there is an electric analog for neuronal membranes. If you want more details, we can always discuss this in the live lectures or through the notes. Cell membranes can be modeled after parallel plate capacitors from electricity and magnetism. If you haven't taken an E&M course, then for specific uses of electric analogs, you can take Physics 224, a course that focuses on the applications of E&M to different biophysical applications. In Physics 380, we'll also be talking about how we can model a cell membrane using more than just E&M. And we'll go through additional examples for parallel plate capacitors as well. So because this is a biological membrane or a neuronal membrane, for example, on one side of the membrane you have lipids, and it can be positively charged. On another side, you can have a negatively charged membrane. And because of that, you can actually model each side as a plate. When you have two plates stacked on top of each other, one that's positive, one that's negative, you end up with a parallel plate capacitor. In between, you do have those lipid tails, so you have to consider a dielectric medium that exists in between the two conducting plates. So what we do is we assume an ion current, or basically a current, flows through a resistor, which would be our protein ion channel, and a dielectric K from our lipid hydrophobic core. So you can actually calculate the capacitance using the capacitance equation for parallel plate capacitors with a dielectric K constant associated with it. That capacitance is equal to 8.8 times 10 to the negative 3 farads per meter squared. You can also calculate the electric field from the surface, which ends up being 1.2 times 10 to the 7 volts per meter. So there are many different things that you can do when you model membranes based off of physical concepts. So last, we want to go through a summary of what you should remember. We did talk a lot about the nervous system and the neurons. You should know about the differences between the central nervous system and the peripheral nervous system. You should also know about different types of neurons and glial cells. We focused a lot on the mechanism of neuronal signaling. So that means the type of potentials that provide a membrane potential, the Danone potential, the Goldman-Hodgkin-Katz potential, as well as the Nernst potential. Should know how action potentials take place and a detailed overview of what happens. Know what plays a role in saltatory conduction, as well as neurotransmission. Then finally, for experimental biophysics, understand how we can manipulate membranes for patch clamp recordings using a pipette, as well as the fact that we can model membranes using physical concepts and different analogs from electricity and magnetism. Basically, how we can model a membrane as a parallel plate capacitor with a dielectric stuck in between with a resistor.